My name is Christine DiDonato, and I'm just about 3 hours south of you in Chicago, Illinois, and I work at the Children's Memorial Hospital Research Center called CMRC, and I work with a group of individuals. I have a basic science laboratory, but we have a team of investigators that we've have developed that are at Children's. We have a new neuromuscular clinician, Nancy Kuntz, who maybe some of you know. She was recruited to Children's from Minneapolis. We have Kristen Kroschel, who is a physical therapist, has worked for many years and been highly involved with families of SMA and their clinical trial outcomes. And Kristen's also at Northwestern. And we're putting together, and I'm a basic scientist, and we've also added in another member, Dr. Debbie Wiesmeyer in the Autonomic Center for Medicine. And the four of us have developed an initiative, and we're going to be adding on other individuals to try and bring and develop a SMA clinical group here. And with Dr. Schroth, hopefully we'll be able to do some very interesting things that I'm going to show on a final 2 slides. And some of this work comes from the basic science that we're trying to translate to the clinical aspects now. So my job for you here today is to give you an update on the basic science research side. So in terms of disclosures, I have no relevant financial conflicts of interest, and I'm not going to be discussing any medications or medical devices. So spinal muscular atrophy is considered to be a neuromuscular disease. So these are any types of disorders that affect your nerves, your muscles, or the interaction between the two, which is called the neuromuscular junction. And so many of the therapies that are ongoing target the survival motor neuron gene, the gene defective in spinal muscular atrophy. But there are also other approaches that we can use that target either the muscle, the nerve, or the junction. So, and what we're going to talk about today is does this show up? Great. So we have a clinical phenotype, which is our SMA, and we know a lot about the genetics, and the gene was cloned in 1995. And so we have where the gene is located at. And so what I'm going to speak to you today is about this whole backside, which is the development of animal models, the basic molecular biology underlying the disease, and therapy development. So I'll go into the basic science aspects into some of the various therapies, and then how we're taking this information from our bench in the laboratory back to the bedside. So all of this is called the bench to bedside process. And so if we speak about spinal muscular atrophy, we all know that it's the second most common autosomal recessive disease, meaning that it requires both mother and father to carry the gene responsible for mutations within the survival motor neuron 1 gene. And 25% of the individuals, the offspring, could be affected. It's clinically heterogeneous, so you can have children with severe type 1, type 2, or type 3, the various forms. And basically the underlying problem is that we have degeneration of the motor neurons. So those are the nerves that come out of your spinal cord and innervate your muscles. And it's caused by low levels of the SMN protein. Now currently we don't have an effective therapy, but what we do know, and this is very important, and you live this every day, is that we know from, in what Kenneth just spoke about, is that early medical intervention is critically important and has dramatic benefits into the overall outcome of our children. And that especially if we take care of the pulmonary function issues as well as nutrition, we can have a major benefit in terms of the overall outcome. So what we're going to break this down today is into 2 parts. I'm going to first talk to you about the basic molecular genetics, the underlying biology of SMA, and from there we need to have that as a basic understanding so that we can begin to understand the various types of therapies that are being developed, because they all target the gene currently that are in process. So I said that the disease is clinically heterogeneous, so we can have are babies that are considered to be type 1, type 2, or type 3, and they can be broken down into various categories. And we won't belabor that point. Now, the question is when we talk about the disease, the defective protein is called survival motor neuron, or SMN. Now, humans, in contrast to other species, such as mouse, or rat, or dog, or cat, we have 2 copies of this survival motor neuron gene, and that's why we actually see the disease in humans. If we didn't have 2 copies of this gene, we wouldn't have this disease. So, and the gene is located on human chromosome 5. And it ends up that after the divergence of mouse to man, right, so of men and of mice, there was this gene duplication. And it ends up that there's only 5 nucleotide changes in the human species that are different, that are critically different between SMN1 and SMN2. And the most critical one is within this exon 7. And this is the last exon that gets translated. So you go DNA to RNA to protein. And so what ends up happening in our SMA children is that the SMN1 gene is lost, but all of our kids have SMN2. Now that's very important because that gives us a natural therapeutic target that we can utilize to develop therapies for. So every one of our SMA babies has an SMN2 gene. It has at least one SMN2 gene. As a matter of fact, the more SMN2 genes you have, the more SMN protein that's made. Okay? So if you have more copies of SMN2, it acts as what we call a dose-dependent modifier. And I'll explain that to you in a moment. So the question is if all of our kids have SMN2, at least one copy, why isn't that enough for our body to sustain our motor neurons, the nerves that innervate our muscles? What's gone wrong? There's something got to be different. Well, it ends up it's that nucleotide in exon 7 that creates that problem, that change in that nucleotide. So this is, if you look at a gene has what we call exons here, here, and here. And this is the critical exon for SMA, this SMN exon 7. And in between, in these yellow lines, are what's called intron. That's the stuff that's removed out. And so what you end up doing is you put exons together, it makes RNA, and it makes your protein that ends up being the effective thing. So in the case of this, now I don't want to belabor this, but what ends up happening is that the regulation of this gene is very complex. So not only do you have the exons and the introns and important information within there, you also have a whole bunch of other factors that come down and bind onto these exons to say, I need to be included or I need to be excluded, okay? And there's lots of them. Now, there's good and there's bad things about that. Having all these extra things also are some of the reasons why we have some of the other drug candidates that are available. Because not only can we target the SMN gene for therapy development, but we can target all these other things that are called splicing factors. Because if we can regulate them, we can also regulate the SMN gene. So what ends up happening? So this is a situation, a little cartoon diagram. If you have SMN1, which has that C nucleotide, what happens is you end up with getting one of these splicing factors. It recognizes this C nucleotide, and it can come down and bind. It brings in all these other proteins. And what ends up happening is you get exon 6 going to exon 7, going to exon 8 in the mRNA transcript, and you get this nice big band, okay? That's what you want. We want the inclusion of exon 7 into our final products, okay? But what happens is our patients don't have a normal copy of SMN1. What they have is SMN2. And instead of having the C nucleotide, they have a T, which in RNA is a U. And what happens is a different protein ends up binding there. And when that protein ends up binding there, this exon isn't recognized very well. And so you don't get some of these other factors that can bind as well. And so what ends up happening is you make only a small amount of this product that contains exon 7, shown here, But you get lots of this other product that lacks exon 7. So many of the therapies that we are targeting all target SMN2 because every one of our kids has at least one copy of SMN2. So if we can find a way to take and block this so that we get exon 7 incorporation in, then we can improve the overall increase of SMN exon 7 transcripts and make a good protein, just like SMN1. And that's what the goal of many of these therapies are. So in a few slides, I'll talk to you about the antisense oligonucleotides. I'll talk to you about a recent publication of Prolactin. I'll talk to you about all these other things. And the entire goal and the whole process that we're trying to do is essentially make more of this in any way that we can, okay? So in the idea here, in the way that we came about this idea that you need to increase SMN levels is both from doing family studies, which all of you have participated in, and where we've looked at our patients and we said, okay, do we have type 1 children, type 2s, or type 3s? So our overall spectrum. And what we did is we asked the question, how many copies of SMN2 do our kids have? And what we found is that those children with a more severe phenotype ended up having fewer copies of SMN2, whereas the children with a milder course of disease had tended to have 3 or 4 copies. And the reason is that each copy of SMN2 produces a little bit of transcript. Okay, so each copy that you have produces a small amount of this. So if you have 1 copy, say it makes 10%, but if you have 2 copies, maybe it makes 20%. If you have 3 copies, 30%. 4 copies, 40%, that kind of thing. So the idea here is just from our previous work of looking working together with our families and in studying SMN2 copy number, we came up with this idea. And that was the basis for understanding and developing therapies that target SMN2. Okay? So this SMN protein, everybody has it. What does it do? Well, it ends up that this protein is expressed in every single cell of everyone's body, okay? And meaning that it's ubiquitously expressed. It's actually a very important protein. It's found in what we call the nucleus, which is where DNA is replicated, as well as out in the cytoplasm. And if we look at a neuron, so this is a neuron, this is the nucleus up here, and this is the nerve, okay? And this is the point that, say, would be your neuromuscular junction. This is the tip, to simplify it. And so what we can find is that not only is SMN located here, in here, but it's also goes along the axon, and it's also at the tip. So we know that SMN gets transported and has roles there. So that's potentially why it has these motor neuron issues. So it's essential for every single cell. It doesn't make a difference whether it's a nerve cell, a heart cell, a kidney cell, whatever. If you don't have the protein, that cell will die. So the question is why is SMA a disease of the motor neuron? And the million-dollar answer is we don't know. What we do know is that what we think is that motor neurons, those nerves that innervate the muscles, have a very high requirement for it. And that in our patients, there's very low levels within the motor neurons. And so we think that that's one of the issues. Now, SMN is what we call a sticky protein. It interacts with a lot of other proteins within the cell. And so therefore, it has a lot of other, not only does it have that function within motor neurons, but it also has numerous downstream targets. That also is a benefit for us because we can think about not only targeting SMN, but we can think about targeting other pathways, okay? And that's one way that, and we'll talk about some of the other therapies that are being developed. So not only can we target therapies that are directly, specifically at SMN2, but we can think about targeting other downstream pathways. So, as I said, it has multiple functions within the cell. one of the basic ones is in what's called splicing. So it's the removal of those introns to push the exons together. And then the other one that it has is within the motor neuron itself. It moves up and down the motor neuron, and it's involved in transporting proteins and cargo and mRNA transcripts down. I love that. I know, thank you. Okay, so I put these in because I didn't know if we would be doing audience participation or not. So I'm going to give a $1 million question. So here's the $1 million question today. It's going to be our Powerball question. And so for audience participation, so you can just raise your hand, A, B, or C. So I've told you so far that SMN is expressed in all cells of the body. It's also required by every single cell in the body because it has this critical role in what we call mRNA splicing. Okay, so it moves and pushes the introns together to generate an mRNA transcript. So the question to you is why do low levels of the SMN protein preferentially affect the motor neurons? Those are the nerves that innervate, go from your spinal cord to your muscle. Is it because, A, we have a splicing defect within the motor neurons? B, is it because the protein SMN has a specific function in motor neurons? Or C, it's both, or you're undecided? So how many think it's A? Okay, how many think it's B? Okay, how many think it's C? Oh, yeah. Oh, you guys are smart, man. Well, we have about 50 that we have to just split $1 million between. That's still good. Okay, so I just want to summarize for you part 1. So SMN1, this gene that has a C nucleotide that normally includes SMN exon 7, is the SMA determining gene. It's this single critical difference in this nucleotide within exon 7 that causes either the inclusion or the exclusion of exon 7 within the mRNA transcript, which then goes on to make a stable protein. Okay? Importantly, exon 7 inclusion, so incorporating exon 7 into the SMN protein, is critical for the function of the protein. SMN2, this copy gene that differs by a single nucleotide, can act as a dose-dependent modifier. So the more copies of SMN2 you have, the milder the disease course. SMN2 ends up being a natural therapeutic target for us because every SMA child has at least one copy of SMN2, and it's expressed in all tissues. Okay, so it's all cell types. It's in heart, it's in nerves, it's in kidney, it's everywhere. Okay? And then the other thing is that SMN protein has many partners that it interacts with, so it has many downstream targets, and so we have the potential for multiple SMN-independent interventions. So essentially, I stole this slide from a colleague, Chris Henderson at Columbia, and in this slide, Chris actually kind of puts it very nicely, is that spinal muscular atrophy is widespread reduction in SMN levels. So in our SMA kids, we have low levels of SMN protein everywhere, in the brain, in the spinal cord, in the muscles, in the heart, the lungs, the liver, everywhere. But it ends up, this low levels, for whatever reason, causes selective degeneration within the lower motor neurons, which are the nerves that go from the spinal cord out to innervate our muscle. Okay? There may be some other cell types in certain individuals that are also affected. Now, the therapeutic strategies for SMA are all targeted towards SMN2. So we have reduced SMN levels. So there's a couple of different ways that we can target this. So I told you that every SMA patient has at least one copy. So if we can increase SMN levels through increasing the total transcription or total RNA, which will then translate to protein of the SMN2 gene, that's fine. If we don't change the total upregulation of the transcript, but we just hit that splicing, that difference in that exon 7 inclusion versus exclusion, so that we promote exon 7 inclusion, that's another option, either through antisense oligos or other drugs that target all those transacting factors. The other thing that we could do that is SMN targeting is to stabilize the protein. Keep that protein longer in the cell. So if it stays around, it can last longer. Every protein turns over, okay? And in a normal situation, the SMN protein, the half-life is about 16 hours. So if we could stabilize that protein and get it to last longer, then we'd have a potential another way of having a therapy. And then the other option is to basically give back SMN to do gene therapy, so we return a normal copy of the SMN gene. Now, if we've missed this, there may be only a window of opportunity to be able to affect these what we call SMN-dependent therapies, which are shown here. But as we move down, we may be able to also block the downstream effects, which are basically the synaptic dysfunction, the misregulation or the deficits between the muscle and the nerve. So if we can maintain those interactions between the muscle and nerve, that's an SMN-independent function, and we may be able to also develop therapeutics based upon that. So I'm going to speak to you about several of these different types of therapies now. But all of these require appropriate animal models. Now, my group, and really what focus my group has been on is developing new models of SMA. I'm not going to speak to those really about today. I'm going to show you one illustration of them, of their use, but we've generated a whole what we call titration series where we could have made new animal models of SMA, of mouse models, that represent severe, the intermediate, and the mild forms. And they don't have some of the issues that some of the other mouse models have. And we're using those to test the therapies to determine how good they are. But all of these require different types. So we have different models that are available that represent the situation, that mimic the situation in human SMA. And there's what we call very severe models that die soon after birth in the mouse. So this is a SMA pup right here, and here's its normal litter mate. And this is about postnatal day 3. This is some people call another severe model. It's a little bit milder here. The average lifespan on this mouse is about 14 days, so from birth to death. And I'll show you a little movie here. So this is, hopefully it'll play. This is the normal mouse. This is its sibling litter mate. And this is the SMA pup. So you can see that. Can everybody see that back there? The lights on? So this is and you can see how he has a little bit of difficulty walking. His little sibling's going to help him get up. Oh, yeah. So these are the models that most people are using right now to test various therapeutic strategies, okay? And the important thing about these animal models are that they mimic the human disease. They show abnormal neuromuscular junctions, and they lose spinal motor neurons, just as our children do. And the problem is that they're great for initial studies, but to have long-term studies, that's where their problem is, because they have a short lifespan, and it makes it a little bit difficult to do some of the drug testing therapies on these. So the therapies, what are they, and who's coming to you soon? Okay, so Kenneth had mentioned Antisense Oligonucleotide splicing correction, which was being taken on by ISIS Pharmaceuticals. So you remember that slide I showed you of SMN2 with all those different proteins aggregated on there? What the idea here is that you're trying to incorporate in Exon 7. There's gene therapy, and there's various pharmacologics, so taking of drugs. And they come in the form of HDAC inhibitors. Some of them are already FDA approved and have already been through clinical trials and are ongoing clinical trials with SMA. There's neuroprotectants, so they're SMN independent but targeting other pathways. And the quinazolines, which are in the phase 1 clinical trial, which was initially initiated by families of SMA and then licensed to Repligen. Now, importantly, here's the important critical thing. It's not that we've pulled these out of a hat and said we're going to test this drug or that drug just to have an idea, just to be testing something and doing something. The important thing here is that each one of these different types of therapy interventions Each of them, importantly, has a mechanistic hypothesis, has in vivo proof of principle, and some safety data that's already available for it. So these are the things that will be coming very soon, hopefully, to clinical trials. So let's talk about the antisense oligonucleotide. It restores normal splicing of the SMN2 gene. So I told you in a normal situation, that slide that I showed with the 0, this is what normally looks like. Low amounts of exon 7 incorporation, high amounts of transcripts that lack exon 7. But when you give this antisense oligonucleotide to SMA mice, you change the splicing ratio to what you want. You want more exon 7 inclusion. And if you vary the concentration of the oligos, you can get increases in SMN. And if you give it to severe models, you can take the survival of those little mice that I just showed you previously, that little guy, from about a 14-day window of survival out to about 26 days, okay? Treating with it. And then here it is in a mild SMA mouse. Now, mice are not the same as men, and for some reason we don't understand this. There's a mild model of SMA, and all the SMA models that are currently published in the mild forms of the disease, for whatever reason, this doesn't work very well. Here it is. You see, they don't have a tail. Now, we don't understand why that is, but they don't have a tail. However, if you give the ASOs, these antisense oligonucleotides, to change a splicing pattern at birth, you can see that these guys do start to grow a little tail. They have their tail's a little bit longer. It's not a normal size of a tail, but it is. So, and so they use this in the mild models. These mild models really don't have a neuromuscular phenotype, but they have this tail phenotype. And in the severe models, you saw the functional deficits, and those are corrected and survival's increased. So currently, the status of this ASO is that it's doing preclinical safety trials, and hopefully the IND filing will be sometime, I believe, in this coming January. And they're gearing up right now and trying to identify sites for clinical trials, is my understanding. So the other one is gene therapy, replacing. So if we can't change a splicing, we can't take a thing, why don't we just give back the gene? So it's called gene therapy. We're going to give back the normal gene. And so it was found by Brian Kaspar's group, along with Martine Barkatz and several other groups, that if you give back SMN in gene therapy very early in the disease course, using an AAV vector, it's a type of virus vector that can transduce and go through the body and hit motor neurons, that you can increase survival dramatically and take that mouse that would die by 11 days of age and have it to live out to over 250 days of age. So from 11 days of death out past 6 months, okay? And you still have them and you see them surviving. Importantly, they also have very good weight gain. They also have very good function. Now, the problem with that is it's a viral gene therapy, so you're giving something back. And so what's going on with that situation with the development of viral gene therapy, it's still going through preclinical studies in monkeys to understand the safety and toxicity of that. And once that's done, then we'll probably go for IND filing to be able to be utilized in clinical trials. And so that's a very long process. Now, what about pharmacological agents? There are some available, such as histone deacetylase inhibitors. These are a type of drug that act upon certain proteins in the body, and it's been shown that they can increase SMN2 gene expression. So that was one of the mechanisms. If you increase overall total SMN2 transcription levels to target SMN protein, you can have a benefit. So FDA-approved drugs already are valproic acid, hydroxyurea, and then this is a preclinical drug that we use in preclinical studies called trichostatin A. So if you take, for example, trichostatin A and you deliver it to the severe SMA mice that you showed, and you provide nutritional improvement as well, you can take a mouse that has maybe a 15 or 16-day window of survival and take it out to about 40 days of age with nutritional, if you incorporate nutritional benefit with it. Now, currently, clinical trials have gone on with valproic acid and hydroxyurea, and those trials are ongoing. And the hydroxyurea, I believe, was done in type 2s or type 1 patients, but both. But those results haven't been reported yet. Now, importantly, the other one is the quinazoline compounds, and the clinical candidate is called RGFP3039. It just initiated phase 1 clinical trials, which Kenneth mentioned to you in July. That's a phase 1 clinical study, so it's about safety and tolerability in normal individuals, so in healthy individuals, so to understand that before it moves into a phase 2 clinical trial. And so this is some work that we did with the preclinical candidate called 495, DCPS, which is a certain type of inhibitor, and targets it targets a protein, this quinazoline compound that my group worked with. And if we treated our mice with this drug, now with this drug, we treated our mice for only 17 days. And I'm going to show you some slides in the next couple of things that can show you what the benefit of this is. So we treated from day 4 to day 20. If you don't treat the SMA mouse with the drug, it'll die by about 20 days of age. However, if you treat it for that short window of time, you'll see what the benefit are here, and the median survival is out to about 87.5 days. So this is another type of drug, that HDAC inhibitor. Again, you can get animals to live out to about 6 months of age when we treat with that. So what do these animals look like? So this is a mouse. Normally we don't have the control mouse to show you how it walks or how sick it is, but because they've died by 30 days of age. So this is a mouse at 30 days of age. This is the control mouse. This is the SMA mouse. You really can't see a difference from it. This is the SMA mouse right here. This is the control mouse. You can see how good its strength is and how good its function is. It has really good climbing ability. Normally the mouse wouldn't be able to hold on there and even hold off, so it has really good strength and function. At 70 days of age, if we look at the mouse, so this is basically 60 days after or 50 days after we stop the treatment of the drug. This is the normal mouse. This is the SMA mouse. Now, if you look closely, maybe you can start to see it has some tremors with it, but it can still walk really well. And if we look 50 days later, that same mouse here, most of the benefit that we had has been lost. So you can see that it's starting to have hind limb paralysis. It really has to use its legs. So what that tells me, and what we didn't know before, is that when we give these drugs, the pharmacological agents, which is different than gene therapy, because if you give gene therapy, when you give it once, it's on forever. With our drug, we gave it for a short window of time. What those results that I just showed you here with this mouse, that shows that drugs can have a time-limited benefit, okay? So that we may be able to dose, stop dosing, and then give repeat dosing. But what's going to need to happen is that continual dosing will probably be an issue. So we know that it's not just one critical window of time, that this is going to be a requirement over a length of time. So that's actually something very important that we didn't know before, and the problem is that the other models died too soon. Even if you treat them with drugs, they still died. And I'm going to explain to you why that was in 2 slides. Now recently, within probably the last 2 weeks, there was a paper published by colleagues from Alex McKenzie's group in Ottawa that talks about prolactin. And prolactin increases SMN expression and survival in a mouse model of SMA via the STAT5 pathway. That was the title of the article. And so essentially what they showed is again, that mouse that I showed you that had this little sibling help him. If they treated the mice with prolactin, they could increase survival by about 10 days, from maybe day 15 to about day 22, something like that. And the way that they postulated that this works through is that you have prolactin, and the prolactin, you treat it, it binds to the prolactin receptor, which then activates a protein called STAT5, which is a signaling molecule. It's part of a signaling pathway. And ultimately, it goes into the nucleus. And ultimately, what ends up happening is you increase SMN2 gene regulation. So SMN, it targets many other genes, but one of the genes that it targets is SMN2. And it increases SMN2 total transcription. So I told you, you can target SMN2, you can target overall transcripts, or that splicing or protein stability. This is a molecule that targets a different type of protein, but the protein binds to the promoter upstream of the gene, activates the gene to make more. And then what ends up happening is you end up increasing mRNA and end up increasing SMN. Okay? And so that's the idea, the working hypothesis on how prolactin increases SMN. Now, prolactin is in theory is very nice because it has several positive things. It's already FDA approved. It penetrates the blood-brain barrier, meaning it crosses your blood-brain barrier, because the target that we need to hit is the motor neurons. The motor neurons are within your spinal cord. So you need to get through the blood-brain barrier. So this drug can penetrate the blood-brain barrier. And importantly, it also improves survival and function in SMA mice. So it fulfills 3 of the important criteria that we look for to develop for clinical candidacy, right? However, there are some negatives, that there's very limited clinical use for that. There is no clinical use at all in SMA kids, so we have no idea what it does, which is critically important. And the other thing is that high levels of prolactin are known to cause side effects. So the one thing that I would caution about is be very careful in its use. People prescribe various things off target, but we don't know how this works. For me, I'm not a clinician, but what I do know is that, for example, hydroxyurea or valproic acid, they have side effects that are very important that maybe don't have effects in other populations, but in our SMA kids, they do, because our SMA kids are different than other populations. So I would caution its use and in overall clinical care. So the one thing that I want to leave you with is that everything that I've shown you only seems to increase survival in these models by about 10 days. It doesn't make a difference what drug it was, unless it was a gene therapy drug. When you looked at these SMA mice, no matter how good or how high the level of SMN protein increase you saw, they only lived for maybe an extra 10 days, an extra 15 days. What does that mean, right? So all these SMN inductive therapies, it means 2 things. There's a short window of opportunity. So we know it doesn't make a difference really whether it's SMA or whether we were talking about cancer or anything else. We know that the earlier the intervention, okay, the earlier the diagnosis, the better the outcome, okay? If we can identify children, our SMA kids, and provide them with better nutritional care, better pulmonology care, better everything, that they will have an overall better course in most instances. It's the same thing with cancer. If you have early detection of cancer, you tend to have a better clinical outcome. SMA is no different. The earlier that we treat the mice, the better the outcome. So there's a window of opportunity for these therapies. Okay, but all the SMA mice are dying. What does that mean? Well, it ends up that, and I'm just showing you some work from our own group. We can induce at postnatal day 0. So as soon as they're born, within 12 hours, we can increase SMN, but we can only double their lifespan. Well, it ends up that what we uncovered with a couple of other groups is that the SMA model mice have slowed heart rates, okay? Now, there's one reason why I bring this up, because families are very concerned that maybe their children have cardiac issues. That may be the case with a subset of children, but we don't know that for a fact. What we do say is that the SMA mice have this issue, And what we're doing here at Children's Memorial Hospital is we're working with a group of individuals and put a team together to actually address that. And that's some of the work that Mary is going to collaborate with us to help us understand better. So when you go to the hospital, you go to the doctor, they put an EKG on you. Well, we do that in our neonate pups. We actually put these little guys from postnatal day 2, we can put them on a little platform with some gel, and we can take their heart rates through their foot pads. And so this is a SMA mouse, and this is a normal sibling littermate. And so you can see the difference in their heartbeat. This is very slow, this is much faster. And when we looked at the hearts of these animals, what we found is that some of the nerves that innervate the heart are actually decreased. So this is a control heart from an SMA mouse sibling. This is an SMA mouse heart. You don't need to know anything except to say, but you can easily see that the difference is these are a little bit thinner, not as long, doesn't have as much branching. This is completely different. So these are part of autonomic innervations, and so we think that there may be some sympathetic or parasympathetic issues, at least in the animal models. And so what we're doing is we've put this and we're developing an actual set of questions and detailed process to understand, and we're looking at this in a very systematic fashion. So in the future, when you see Mary or if you come to Chicago, you may be asked if you would like to participate in some at-home monitoring systems, which is using a device that will actually take your EKGs and monitor the breathing and respiration and look at this. So we're trying to do this in a very clinical, very systematic fashion. So we don't know what the answer is. We know that it occurs in mice, but that certainly does not mean that it happens in the humans. And what we're trying to do is understand what, if it does, and if so, what is the occurrence, and how can we use what we know in terms of our clinical care to improve overall health and function. So this is our team at Children's Memorial Hospital. This is the research institute that I work with. I mentioned Nancy Kuntz. Nancy is our neuromuscular clinician who's been with us for about a year. Kristen Grischel is a physical therapist. She's highly involved in clinical outcomes and clinical trials. Myself and Debbie Wiesmeyer, who's in autonomic medicine. And so in the summary for the second half is I told you about widespread reductions of SMN levels triggers selective degeneration of those nerves that innervate your muscles. And our current understanding of SMN biology has led to the identification of molecular targets for therapeutic development. And I've went through and I described several of those different types of possibilities to you. And importantly, all of the therapies currently that are moving towards clinical trials have are mechanistically based on SMN biology, and they're rapidly approaching the clinic. And the window of opportunity to affect a benefit for SMN therapies in severe mice is time limited. However, what we don't know, and this is another important critical question that I did not actually bring to the forefront, the time-limited window in SMA mice does not necessarily mean that it's such a short window of opportunity in our SMA kids. So this is the difference between mice and of men, and this is where we need to look at it and understand. Certainly, the earlier the intervention, probably the better the outcome. And so the challenges that we face as basic scientists and clinicians, as well as families, are translating all these wonderful results that we have preclinically and in these models to actually translating them to the clinic. And so the only way that we're going to be able to do this is by sound clinical trial design through the support of families of SMA whose understanding is giving towards research to support basic clinical trial development and outcome measure development. Our improvement of our standards of care. We need to have our kids be as healthy as possible and have the best, and everybody have a common standard of care so that everybody is on the same playing field so that we don't have differences in our care. So those are very important aspects. So I'm going to leave you with the second $1 million question. So this is the $100,000 Powerball. So I told you that SMA is caused by homozygous loss or mutation of the SMN1 gene, and all SMA patients have at least one copy of SMN2. So which are the following are true? SMN2 has the potential to replace SMN1, but is not expressed in the correct tissues. Number 2, SMN2 produces small amounts of functional protein. That should be 3. Or 3, SMN2 copy number increases. Generally, SMA disease severity decreases. So how many people think it's A, 1, 2, and 3? How many people think it's B, 2 and 3? How many people think it's 1 and 3? 1 and 3. We didn't have that much audience participation. Only a couple people go win. It's B, 2 and 3. Okay. And with that, I think I will end and just acknowledge the many people from many laboratories who've worked with us. Thank you.